I'll be telling you about our work on hepatic glucagon signaling, INSP3R1 as the conductor. I have no conflicts of interest that are relevant to any of the data or the topics that I'll be discussing in this talk. Because this symposium is marking the 100th anniversary of the discovery of glucagon, I wanted to lead with Banting and Best and John McLeod, who was the physiologist in whose lab the initial experiments were done. Their team were injecting diabetic dogs with pancreatic sludge, trying to prove that a factor that was derived from the pancreas could be used to treat diabetes. And they made the initial observation that injection of the pancreatic sludge could cause a quick rise in blood glucose before it fell. Now, they dismissed this as being the result of epinephrine, so a stress response to injection of the foreign material, and it was not until later that Merlin and Kimball confirmed the existence of a glucose-raising factor in pancreatic extracts, and they named it glucagon, denoting glucose agonist. Since glucagon was discovered 100 years ago, we have to have some idea of what it does in the liver, right? Well, conventional wisdom holds that when glucagon binds to its receptor, it has two main actions on liver, both related to increasing hepatic glucose production. First, it activates glycogen phosphorylase and thus stimulates glycogenolysis or glycogen breakdown. And secondly, it increases transcription of gluconeogenic enzymes, therefore increasing gluconeogenesis. But one caveat to this is that glucagon can increase hepatic glucose production very, very rapidly, which makes sense for a counterregulatory hormone. Here you can see data from Alan Sherrington's group showing that in dogs, an infusion of glucagon can increase hepatic glucose output within just 15 minutes, which obviously is a time frame inconsistent with transcriptional regulation telling the whole story. So this is an idea that we'll explore at length in the data that I'm going to tell you about today. So let's go into a little bit more detail about putative mechanisms, not just actions, of glucagon to acutely stimulate hepatic glucose production. So it's long been believed that when glucagon binds to its receptor, which is a 7-transmembrane GPCR, it activates the adenylate cyclase cyclic AMP pathway, which increases pKa activity. Also, independent of the cyclic AMP pathway, glucagon also activates phospholipase C signaling. And together these signals converge not only to increase transcription of gluconeogenic enzymes, but also to phosphorylate the inositol triphosphate, or IP3 receptor. And the IP3 receptor signals on the endoplasmic reticulum to release calcium. Calcium is released into the cytosol, and it enters the mitochondria through mitochondria-associated ER membranes, ultimately through a mechanism that had been something of a black box, stimulating hepatic glucose production. But despite this textbook mechanism, there is an outstanding question which we've attempted to answer in recent years. And that is, what is the mechanism by which glucagon stimulates hepatic glucose production? So the first fundamental driving question of the studies I'm about to show you was simply, does IP3 receptor signaling have a role in glucagon regulation of hepatic gluconeogenesis? And if so, how? From many previous well-done studies, there's no doubt that glucagon can increase transcription of gluconeogenic enzymes, most notably G6P and cytosolic pepsi-K. But on the flip side, we know that glucagon can stimulate hepatic glucose production within minutes, which is a time frame incompatible with transcriptional regulation telling the whole story, but is why we use it as a rescue treatment for hypoglycemia in insulin-dependent diabetes. So we wanted to explore this question further. To examine the role of hepatic IP3 signaling in glucagon's action to stimulate gluconeogenesis, we studied mice with a liver-specific knockout of the main inositol 3-triphosphate isoform that's found in the cytosol in hepatocytes, that is IP3R1. So IP3R2 is also found in hepatocytes, but it's primarily concentrated in the ER rather than in the cytosol. And since, as you know, the cytosol is the home of the gluconeogenic machinery, we chose IP3R1 as the isoform of interest to study glucagon regulation of gluconeogenesis in this study. So here you can see validation of the model from our colleague, Dr. Barbara Ehrlich, who obtained these mice as a kind gift from Dr. Andrew Marks at Columbia. So here you can see that IP3R1 is robustly knocked down in the liver of these knockout mice. So we first showed that wild-type mice respond to glucagon by increasing calcium signaling in hepatocytes by around 50%. However, this is significantly blunted in IP3R1 knockout animals, which exhibit a 70% reduction in their mitochondrial calcium response to incubation in physiologic concentrations of glucagon. Next, we wanted to understand whether and how glucagon stimulation of gluconeogenesis would be affected by the absence of mitochondrial calcium signaling. To that end, we performed a 2-hour infusion of glucagon in awake mice, and we found that whereas glucagon produced a modest but significant increase in both plasma glucose and insulin concentrations in wild-type mice, it had no acute effect in IP3R1 knockout animals. Next, we asked, what is the mechanism for the glucagon-induced rise in plasma glucose concentrations? Well, similar to the rise in plasma glucose, we detected a small but significant increase in endogenous glucose production both in vivo on the left and in vitro on the right in wild-type animals, but not IP3R1 knockout mice. I think the in vitro data are particularly important because they dissociate any changes in plasma glucose from changes in plasma insulin, since the media used for these in vitro studies did not contain any insulin. Under these conditions, 18-hour fasted mice or serum-starved hepatocytes, the increases in glucose production in vivo and in vitro on the previous slide represent increases in gluconeogenesis, not increases in glycogenolysis, because these mice and these cells are glycogen depleted, and glucagon does not alter hepatic glycogen content, as you can see on this slide. So any glucose appearance that we see must come from gluconeogenesis. Even if that 15% reduction we see in liver glycogen with glucagon treatment in the wild-type mice were significant, which it isn't, the rate of net glycogenolysis that we can calculate from this would be just 5% of the total glucose production rate. So that means we can confidently say that glycogenolysis is minimal under these conditions. And the changes we see in glucose production are a result of increased gluconeogenesis stimulated by glucagon and not a result of increased glycogenolysis. So, having convinced ourselves that glucagon acutely stimulates endogenous glucose production by a calcium-dependent process in hepatocytes, we next asked, how is this happening? So we applied a method we developed called positional isotopomer NMR tracer analysis, or PINTA, which allows us to measure substrate contributions to glucose production. So that you can understand what we're doing here, I'm going to quickly walk you through the gluconeogenic pathway. So if we start with pyruvate, it can enter the TCA cycle in 2 ways. Number 1, through pyruvate dehydrogenase into citrate, or number 2, through pyruvate carboxylase into oxaloacetate. Many of the gluconeogenic amino acids enter the pathway this way, either into pyruvate, so that would be alanine, cysteine, glycine, or serine, or entering into the TCA cycle. That would be threonine, tyrosine, phenylalanine, glutamine, and others. So oxaloacetate makes PEP through PEPCK, and PEP is converted to glucose after a couple more steps through the gluconeogenic pathway. So it's here at the dihydroxyacetone phosphate GA3P step that glycerol can enter the gluconeogenic pathway. It simply pours through, largely unregulated. So we can really divide up gluconeogenesis into what happens north of PEP and what happens south of PEP. So what we measure using mass isotopomer distribution analysis, which is what PINTA relies on, is gluconeogenesis from PEP versus gluconeogenesis from glycerol. And when we do that analysis on our livers from the in vivo PINTA studies, from which I showed you endogenous glucose production earlier, we find a 30 micromole per kilo per minute increase in hepatic gluconeogenesis from PEP and everything that comes before it, including amino acids. That increase we see in wild type, but not knockout mice treated with glucagon, which is approximately equal to the total increase in total glucose production in wild type mice after glucagon treatment. The question then becomes, how does glucagon increase hepatic gluconeogenesis through IP3R1 signaling? Well, first we wanted to validate that we see the same thing that others see with regard to the key role of phospholipase C protein kinase A signaling in glucagon action. In examining the role of phospholipase C pKa signaling in hepatic glucagon action, let's start with pKa. We did in fact observe an increase in liver cyclic AMP and liver PKA activity in both wild-type and knockout mice. Let me just remind you here that it's not surprising that we saw the same induction in cyclic AMP and PKA signaling in wild-type and knockout mice because PKA is upstream of IP3-mediated calcium signaling. So that just tells us that the input signal to calcium signaling is the same. And we then validated the importance of this in hepatocytes. Whereas glucagon increased endogenous glucose production, inhibiting PKA did nothing under basal conditions, but it blocked 100% of the effect of glucagon to stimulate hepatocyte glucose production in wild-type mice. In contrast, in IP3R1 knockout mice, in which glucagon had no effect to stimulate glucose production, we saw no effect of PKA inhibition. So glucagon didn't stimulate glucose production, but inhibiting PKA did nothing further. So that, to us, validated that PKA signaling acts through calcium, at least with regard to stimulation of glucose production. Next, we applied two phospholipase C inhibitors, ET18-OCH3 and U73122 in hepatocytes. We found that, like PKA, inhibiting phospholipase C did nothing under basal conditions, but blocked 100% of the effect of glucagon to stimulate hepatocyte glucose production in wild-type mice. In contrast, in IP3R1 knockout mice, in which glucagon had no effect to stimulate glucose production, we saw no effect of phospholipase C inhibition, validating that, like PKA, phospholipase C signaling acts through calcium to stimulate endogenous glucose production. We know from our calcium data and from others' work that IP3 receptor signaling causes increased intracellular calcium release, and we first wanted to verify whether this plays a functional role in the liver's response to glucagon, at least in terms of stimulating hepatic glucose production. So first we measured liver CAMK2 phosphorylation by Western blot, and we found that glucagon causes liver CAMK2 phosphorylation in livers of wild type, but not IP3R1 liver-specific knockout. What impact does this have on hepatic glucose production? Well, we started by measuring glucose production in hepatocytes, and we again saw that glucagon promoted hepatocyte glucose production, and there was no effect of inhibiting CAMK on basal glucose production in hepatocytes that were not stimulated with glucagon. However, the CAMK2 inhibitor completely blocked the ability of glucagon to stimulate hepatocyte glucose production, showing that calcium signaling is required to mediate glucagon's effects on hepatic glucose production. In contrast, there was no effect of the inhibitor in IP3R1 knockout hepatocytes, consistent with the idea that IP3R1 exerts its effect on hepatic glucose production through calcium signaling. So what is the mechanism by which glucagon stimulates hepatic gluconeogenesis through calcium signaling? Well, it does not appear to be transcriptional because gluconeogenic protein expression data are dissociated from the effects of glucagon on endogenous glucose production in vivo. Although we see increased pyruvate carboxylase, i.e. gluconeogenesis from PEP, we actually see downregulation of enzyme expression, particularly in the IP3R1 knockout animals. So what are the other potential regulators of gluconeogenesis? In addition to transcriptional regulation, which we've ruled out here, we also have to consider substrate level regulation. One of the most direct gluconeogenic substrates is glycerol, which pours into glucose in a largely unregulated manner. And finally, we have allosteric regulation. Acetyl-CoA, the end product of beta oxidation, is known to allosterically stimulate pyruvate carboxylase activity and thus gluconeogenesis. And indeed, when we measured liver acetyl-CoA concentrations, we found that glucagon increases acetyl-CoA content in wild type, but not IP3R1 knockout mice, which could be the explanation for the increase in hepatic gluconeogenesis from PEP that we see in animals and hepatocytes treated with glucagon. Next, I want to remind you how acetyl-CoA is regulated. When lipolysis occurs, fatty acids are released, as well as glycerol. The process of beta oxidation breaks down fatty acids 2 carbons by 2, ultimately forming the 2-carbon acetyl-CoA. So this emphasizes that the easiest way to have increased acetyl-CoA is to have increased lipolysis. And indeed, we found that glucagon increased the release of both glycerol and non-esterified fatty acids in isolated hepatocytes from wild type, but not IP3R1 knockout mice. So these data suggest a liver-specific calcium-dependent effect of glucagon to promote intrahepatic lipolysis, thereby increasing hepatic acetyl-CoA. To directly test the possibility that calcium-driven lipolysis may be required for glucagon's gluconeogenic effects, we treated mice with an adeno-associated virus to knock down ATGL, a key rate-limiting lipolytic enzyme, in a liver-specific manner. Liver-specific ATGL knockdown lowered liver long-chain acyl-CoA and acetyl-CoA concentrations in wild-type mice following glucagon treatment to those measured in the IP3R1 knockout mice regardless of glucagon, consistent with an effect to reverse glucagon-stimulated increases in intrahepatic lipolysis. Suppressing lipolysis in a liver-specific manner totally prevented the ability of glucagon to raise plasma glucose or insulin concentrations. And it did so by preventing glucagon's ability to stimulate endogenous glucose production or gluconeogenesis from PEP in wild-type mice, demonstrating that glucagon's ability to acutely stimulate gluconeogenesis does indeed require calcium-dependent stimulation of intrahepatic lipolysis. So to summarize what I've shown you thus far regarding the mechanism by which glucagon stimulates hepatic gluconeogenesis, we found that upon binding to its receptor, glucagon stimulates adenylyl cyclase-mediated cyclic AMP pKa signaling, as well as activating phospholipase C. Together, the actions of these cascades converge on activating IP3R1 signaling to release calcium into the cytosol and through mitochondria-associated ER membranes into the mitochondria. The increase in cytosolic calcium causes increased CAMK2 phosphorylation and increased ATGL-dependent intrahepatic lipolysis. By supplying more fatty acids to the oxidative machinery, this increases mitochondrial acetyl-CoA concentrations, which allosterically activates pyruvate carboxylase. Also due to the increase in lipolysis, more glycerol is simultaneously supplied to the hepatocyte cytosol. And together, these increases in pyruvate carboxylase activity and glycerol concentrations increase hepatic gluconeogenesis in glycogen-depleted mice. Based on the known ability of glucagon to activate hepatic glucose production, glucagon antagonists and glucagon receptor antagonists have become a promising class of therapy for diabetes. Glucagon antagonists or receptor antagonists clearly lower plasma glucose and A1C in people with type 2 diabetes. However, they show a consistent adverse effect of promoting weight gain, hepatic steatosis, and hypertriglyceridemia, all of which, of course, are especially concerning in the population with type 2 diabetes. On the flip side, glucagon and GLP-1 dual agonists have also gained a lot of attention. They cause weight loss, reverse NAFLD, and improve glucose tolerance, with some GI side effects being really the only major adverse effect of this class of drug. So these data certainly imply that there may be an effect of glucagon to stimulate lipid oxidation and an effect of glucagon antagonism to limit lipid oxidation. We wanted to better understand this phenotype. And so we asked, is glucagon's effect to promote lipid oxidation mediated by IP3R1? So as you know, fat accumulation in tissues occurs very simply due to an imbalance between influx and outflux of lipids. To examine why that might occur, we measured rates of oxygen consumption in hepatocytes and found that a physiologic concentration of glucagon caused a 30% increase in oxygen consumption rates in primary hepatocytes from wild-type mice, but it had no effect on IP3R1 knockout hepatocytes, indicating that this effect of glucagon to stimulate mitochondrial oxidation is also calcium dependent. When we performed PINTA studies to measure in vivo mitochondrial oxidation rates, we found that glucagon induced a 4- to 5-fold increase in mitochondrial citrate synthase flux, which could be attributed almost entirely to increases in fat oxidation in vivo in wild-type mice, but it had no impact on citrate synthase flux or fat oxidation in IP3R1 knockout mice. So these data, we think, could potentially explain the clinical observation that patients who are treated with glucagon receptor antagonists are prone to develop NAFLD and transaminitis. Based on these data, showing that glucagon acutely increases mitochondrial oxidation, as well as the clinical data showing NAFLD in folks treated with glucagon antagonists, we hypothesized that chronic glucagon treatment would reduce ectopic lipid concentrations and improve hepatic insulin sensitivity in rodents with diet-induced obesity. So we took 8-week high-fat fed rats and infused them with glucagon for 10 days to quadruple plasma glucagon concentrations. This intervention more than doubled hepatic citrate synthase flux, reflecting an increase in hepatic mitochondrial oxidation in response to glucagon treatment. Increasing hepatic mitochondrial oxidation reduced liver triacylglycerol and diacylglycerol concentrations by 50% or more, and reduced PKC epsilon translocation by more than 85% in liver after just 10 days of treatment in these obese animals. These effects, importantly, occurred without any differences in food intake or body weight, so you don't have to induce nausea to get these beneficial effects. In addition, chronic glucagon lowered plasma glucose and insulin concentrations throughout a glucose tolerance test, demonstrating a marked improvement in glucose tolerance in these animals. But of course, these data simply show that chronic glucagon treatment can reverse NAFLD. They don't prove whether or not this effect occurs through IP3R1 action. So finally, we performed this study in wild-type and liver-specific IP3R1 knockout mice. When we treated with glucagon by subcutaneous pellet for 4 weeks, we observed a 3-fold increase in TCA cycle flux and in mitochondrial fat oxidation in wild-type mice, but there was no effect of glucagon on oxidation in IP3R1 knockout animals. Chronic glucagon treatment reduced liver triglyceride and diacylglycerol concentrations by 30% or more, and reduced PKC epsilon translocation from the cytosol to the cell membrane by more than 80% after 4 weeks of treatment. And again, these effects occurred without any differences in food intake or body weight. And perhaps most importantly, particularly when addressing an audience who think a lot about diabetes, chronic glucagon treatment lowered plasma glucose and insulin concentrations throughout a glucose tolerance test in wild-type mice, but it had absolutely no effect in IP3R1 knockouts, demonstrating a marked improvement in glucose tolerance with glucagon treatment that is dependent on IP3R1-mediated calcium signaling. So to summarize what I've shown you throughout the talk regarding the mechanism by which glucagon stimulates hepatic gluconeogenesis and fat oxidation, we found that glucagon stimulates the pKa and phospholipase C pathways to activate hepatic IP3R1 signaling, calcium release, and intrahepatic lipolysis. By supplying more glycerol and increasing hepatic acetyl-CoA, this promotes hepatic gluconeogenesis. At the same time, by supplying more calcium to the mitochondria, IP3R1 signaling promotes mitochondrial dehydrogenase activity, leading to increased mitochondrial fatty acid oxidation and reducing hepatic lipid. So taken together, we can conclude that glucagon acutely increases hepatic gluconeogenesis by stimulating intrahepatic lipolysis and increasing hepatic acetyl-CoA concentration through IP3R1 signaling. And that by increasing rates of hepatic mitochondrial oxidation, short-term glucagon treatment may be an attractive therapeutic approach to treating NAFLD-associated hepatic insulin resistance. And where do we go from here? Well, since opening my lab in 2018, we've been focusing on using approaches to increase hepatic mitochondrial oxidation to improve insulin sensitivity and outcomes in obesity-associated cancers. Colon cancer is a good example of this. We see here that feeding mice with a high-calorie diet accelerates colon cancer growth. However, by treating mice with a mitochondrial uncoupler to increase hepatic mitochondrial oxidation and burn liver fat, we're able to reverse insulin resistance and slow colon cancer growth rates to what we measure in chow-fed lean animals. This effect is insulin dependent, as evidenced by the fact that when we treat with insulin by subcutaneous pellet, we increase tumor growth rates to what we measure in untreated obese animals. And we can see similar phenotypes in breast cancer. Obesity accelerates breast cancer growth, but we can slow it to rates measured in lean animals by treatment with a mitochondrial uncoupler. This effect is insulin dependent because when we treat with insulin, we accelerate growth rates to what we measure in the obese animals, and the uncoupler is no longer effective. So we believe that mitochondrial uncoupling holds promise, not only in reversing NAFLD, but potentially also in slowing obesity-associated insulin-dependent tumor growth. And with that, I'd like to close. I'll acknowledge many collaborators. My longtime mentor, Jerry Schulman, in whose lab most of this glucagon work was done, as well as the members of my lab today and our collaborators, including Mike Nathanson, Barbara Ehrlich, and members of their lab, Ali Brill and Matthias Guerra, without whom this work would not have been possible. I thank you so much for your attention. Again, I'm very sorry I can't be there, but I appreciate your listening.